My name is Kajatu Padiga. I'm a second year general sciences major.
I'm Hyunbin Gang. This is my third semester and my major is biology.
And our project was is Progurt 2018, and our mentor is Dr. Murti. So why did we research yogurt? Our mission originally was to quantify the amount of microorganisms in fresh-made yogurt and store-brand yogurt and some fermented foods. We anticipate that the fresh-made yogurt would have more probiotics in it than the store-bought. So in order to quantify the microorganisms from this, we used the serial dilution factor method. To understand our project a bit, I will give you a brief background on what the human microbiome is. It's a research initiative that started in 2008 by the National Institutes of Health, where their purpose was to identify and characterize the human microbiome on 5 major sites, which were the nose, the mouth, the urogenital, the gastrointestinal, and on the skin. And what they found was that there are over 100 trillion microbes in and on the human body, which means that there are 10 times more microbes or bacterial cells than human cells. So 95% of them live in the GI tract, and most humans are susceptible to diseases from the GI tract, and a majority of the diseases that are in the human body are linked to the GI tract and these microbes. So probiotics are commonly known as the good bacteria. What we know about them is that they aid in the synthesis of vitamins, they suppress pathogens, and they enhance the immune system. So probiotics usually colonize the gastrointestinal area, which are usually in the intestines, and they may improve conditions like IBS, Crohn's disease, and diarrhea. And they're commonly found in fermented foods such as yogurt and kimchi and kombucha. And the most widely consumed of these is yogurt. So there are many different types of bacteria, but the 3 major types that are found predominantly in the GI tract are the lactobacillus, the Bifidobacterium, and the Saccharomyces boulardii. And the bacteria that we focused on in our project is the lactic acid bacteria, which is most notably the lactobacillus. In order to be considered a probiotic, there must be an adequate quantity, which is between 1 and 10 billion CFUs. CFUs are colonies that form from a particular bacterial cell. So we assume that each CFU comes from a viable bacterial source.
So this is how we do dilution. And what we do is basically different from all these amounts. We take 500 microliter of yogurt and then transfer it to the first test tube to make 10 to the -1 dilution. And then we repeat the process until we dilute up to 10 to the -5 or 6 or any factor that we need to make. Then we transfer small amount of solution, each solution to the plate, and then we incubate them for 2 or 3 days to see the bacterial growth. This is how we count with the markers, and after we count them, we calculate the CFU using this formula. In this semester, we are doing experiments with 3 types of yogurt: Dr. Murti's Indian yogurt, and Sadiq's Greek yogurt, and Adele's Albanian yogurt. During the last 2 weeks, we diluted them as we saw the bacterial growth. For the Indian yogurt, we saw most colonies on 10 to the -1 plate among the 3 plates that we diluted. And the CFUs of Indian yogurt were 4.6 times 10 to the 3. And for serving size, which was 150 milliliter, there were 6.9 times 10 to the 5 bacteria. For the Greek yogurt, same as Indian, we saw most colonies on 10 to the -1 plate, but we couldn't see many colonies from the Greek yogurt. But we made made a fresh Greek yogurt yesterday, and we're going to dilute it again. Hopefully this time we can see more colonies. For Albanian yogurt, there were so many colonies that we had to dilute it again to the lower dilution factor. Even with the lowest dilution factor that we have here, which was -3, it was hard to count them all because there were so many colonies. These are graphs to compare the results of 3 types of yogurt.
We had a few drawbacks during our research. We experienced like a long period of where there was no growth with the Greek yogurt, as Human mentioned. What this means is that the bacteria wasn't able to grow in the media that we had. Because we were able to make fresh yogurt, we do know that there was bacteria in the sample. So we just, came to the conclusion that we couldn't reproduce the same growth conditions within the lab. And maybe there's more diversity within that yogurt that we're just not able to identify with the media that we are provided with. So we made some modifications. Instead of using LB plates and TSA plates, we used MRS plates, which are specific to lactobacillus. And they have like yeast extraction in them, where there's reduced oxygen and it increases, the extraction helps in the increase of CO2 levels. So we're expecting that there will be an increase in lactobacillus growth. And in addition, we hope that there'll be secondary accompanying growth where we'll see some diversity as well. So what's next? We're gonna try a different enumeration technique. Instead of using the LB and the TSA plates, we might use the membrane filtration. And after that, we'll be able to see if there's any diversity within the fresh yogurt, which will entail doing PCR, sequencing, and DNA extraction. And we encourage you all to provide us with some samples of fermented foods from wherever you're from. We're Turkey, Middle East, I don't know where you all are from, but we'd love to have your samples and able to do some research on them.
So these are the contact and blog information if you just want to contact us. And
So we'd like to thank the biology department, our mentor, Dr. Murthy, our lab technician, Richard, and the entire STEM program for providing us with this opportunity.